Sarah start with a welcome to country before I go on to the introductions and the actual talks.
Yeah, so I asked to do the welcome to country, acknowledgement of country, not the welcome. So we are part of ANZUC, so we look after Australia and New Zealand. So we actually look after 19 universities around these, the two countries, and we do represent a broad community. So I definitely wanted to have my acknowledgement of country. I do acknowledge that we are Ngambri and Ngunnawal peoples nation right now, but we do represent a lot more and definitely see country. And definitely for the geoscience space, we love this interaction too. I do want to acknowledge that that our Indigenous people, it's amazing to work with them. They have such a great knowledge of definitely climate change, sea level rise. A lot of our First Nations people know what to do if a tsunami comes. I don't think most of our population knows what to do when that happens. They have history of volcanic eruptions. It's really amazing. And just click. And I also just wanted to highlight a great expedition that's coming up. So we send students out onto the research vessels too to learn STEM skills at sea. So we're actually sending 8 First Nation people from Australia and New Zealand out to Italy. Then they go on a ship to Amsterdam to learn some amazing skills. So just wanted to highlight that. That's going to happen in April. So please watch this space. Get back to you.
Thank you, Sarah. So Sarah Kachovich is also going to be our first speaker of this afternoon. Sarah is a superstar of STEM. STA in Australia has established the superstars that you might have heard of, and fantastic that you're part of that program. She's based at the ANU here in Canberra, and she's the director of the IODP, Deputy Director of the IODP. So she's working on zooplankton in the ocean, and she will give the first talk for this afternoon before we go over to YouTube. Very much forward.
Did you want to introduce the other speakers?
Should I introduce all the speakers now? I will do that. The second speaker is Gordon Southam. He's professor in the School of Environment at the University of Queensland, with previous appointments at the School of Earth and Environmental Sciences and the School of Earth Sciences, and as a Bailey UQ Chair in Geomicrobiology. Before Australia, Professor Southam was a Canada Research Chair in Geomicrobiology at Western University. After that, we will be hearing from 2 presenters at the University of Tasmania. And Linda Armbrecht is a paleogeneticist and ARC DECRA Fellow at the Institute for Marine and Antarctic Studies at the University of Tasmania. And her research focuses on the reconstruction of marine ecosystems using novel sedimentary ancient DNA techniques, with an emphasis on the Antarctic environment, where research into resilience and sustainability is critical due to climate change. And our fourth speaker will be Lucinda Duxbury, who's a PhD student at UTAS, exploring how ancient DNA and RNA preserved in seafloor sediments can help us reconstruct past marine ecosystems in the Arctic and Antarctic. And in June, she'll be trading in the Tassie winter for an Arctic summer. Very nice. Sailing as a microbiologist and geochemist on the first voyage of IODP's research vessel, the JOIDES, that's how you say it, Resolution. They're all very envious. So it would be an interesting program this afternoon. Could you please keep your questions to the end so that we don't cut into people's times, and there'll be a question and answer for everyone at the end. So thank you, Sarah, and we look forward to your talk.
Thank you. Great introduction. So I'm going to give a quick brief overview of what IODP actually is and scientific ocean drilling. I'm also going to give a very quick look into the greatest hits of the deep biosphere, so you can really get a quick taste of what we've been doing, because we really don't socialize enough. Definitely as earth scientists, we're a very interdisciplinary program, very international as well, as you'll soon find out. And I don't think we do a well enough job in Australia and New Zealand, and there are so many opportunities, and we want to make sure that you guys are aware of it and know of your opportunities here. So I've created this little graphic just to quickly demonstrate what the International Scientific Ocean Drilling Program. So we are drilling the deep sea. We're the only international program that actually does this, and we go very deep, as you'll soon find out. So I've got this little core here. So you can come have a look. This is actually the day the dinosaurs died. So it's very cool to have a close look into that and actually unravel what just happened here. And go, definitely we drilled Chicxulub crater 2016, and you can go second by second into what happened. But with these cores, they are like the libraries of our oceans, and we recover these books, these cores, and they tell us about the past history. But in them, we also collect sediments, rocks, fluid, and microorganisms are the kind of 4 key things we're collecting. And at the bottom there of my slide, you can see that our main objectives of the next 25 years are these themes down here. So we're going to focus a lot of this presentation for this next hour on the first 2, but we do a broad range. So if you are interested in any of these kind of topics, IODP has a spot for you. So IODP, the Scientific Ocean Drilling Programs, it is plural. We have been a continuous program, but with different names. We've been going for 56 years. I think we're a little bit older than NASA. That's our claim. There we go. And these are the current members of the dark blue. And as you can see, Australia and New Zealand are the only ones in the Southern Hemisphere. It's a bit of a problem, but there's still a very international flavor, and we've had previous members and observers as well sail and contribute to IODP science. So truly international, very sensitive. And kind of the timeline of the programs, we use a lot of acronyms. We play a lot of bingo when we give talks. So you might hear names like DSDP was the first iteration in the 1960s, then ODP, then IODP, then IODP again. We took away the drilling to say discovery. We'll be bringing back drilling for the next IODP 3. What would science be without its acronyms? So I'll just check the chat. Oh, sorry, I just got to change my viewing slide. I'll stop sharing and share. Thank you. How is that online? Can I have a, oh, good. Thank you. So how do we drill? So we have sort of 3 main platforms. We hire vessels to go do shallow kind of coastal regions or very bespoke kind of drilling, or we have these 2 deep water vessels currently. So we have the Chicxulub, there's a little model down the front there if you want to have a look at specs. And we also have the Geordie's Resolution. So I'm not going to go into detail too much how it's done, but here's just a quick schematic. It is blue water drilling. So we have a giant vessel there. We have a bunch of thrusters. We have a giant water column up to many kilometers. This one here is the Chicxulub, so it's actually a riser driller. So this part here is actually the riser. It anchors itself to the bottom, and it can go very deep and handle fluid pushback. The difference is the Geordie's Resolution is a little bit smaller, cheaper, and it's riserless. So it doesn't have this operation here. It can go in deeper water, but it can't drill as deep, but it's still about 2 kilometers it can drill. So it's still quite deep. So it can be mobilized very easily. So it can go all around the world. So we do have a map here too. All those dots are where we've drilled. You can say where you want to drill as well for those in the room. And you can see that we've actually covered a lot of the ocean. So that's all we're going about the drilling. Where do we drill? So here's a map I've created out of the Spillerhouse map projection. If you haven't seen this before, I think it's an excellent way to demonstrate the world's ocean. It really demonstrates how the fish see the world, how it's all connected by one ocean. So you have Antarctica in the middle. Sorry, for those online, gotta, whoop, spoilers right there. You have Antarctica in the middle, you have Australia here. New Zealand's off the side here. This is the Pacific Ocean. You've got the ocean currents going around too. Indian Ocean, India, Africa. There's the Atlantic Ocean and the North Pole up here. So you can actually see some big misconceptions on this kind of map projection that the North, kind of the Arctic, the North Pole is actually landlocked, so it does behave very different. And I'm going to say Antarctica is the heartbeat of the ocean, really does drive. And it should really demonstrate the importance of the Southern Ocean, definitely around us. So all the yellow dots are where we've drilled before. all the blue dots are the kind of proposed drill sites for the future. So hopefully we'll see a lot of drilling in our area. So we've done about 462 kilometers. Sorry, I'm pregnant, so I get excited, talk too fast, get out of breath. So why do we need to drill to sample the deep biosphere? So we have a lot of life. We know the water column is full of life, but actually the largest biosphere is actually predicted to be in the subsurface. So life in the ocean extends way below the subsurface. So we do two types of drilling into the subsurface. We drill down or we do boreholes. So we do a borehole and we actually install borehole observatories over long term. So for a program that is 56 years old, it's the perfect place to do long-term experiments. I think a lot of programs we see around the world are very short, and it's really hard to do these long studies, but in this program, it's absolutely essential, and it's a very nice place to do long-term studies and have that long-term funding, which is always handy. So, and the subsea floor environments span high pressures, extreme heat, exceeding low carbon availability, and nutrient stress, but yet we find life everywhere. About 20 years ago, we actually found life where we shouldn't have found life, and definitely our NASA colleagues, all of a sudden, we're like, you're finding a life where? And they started to work very collaboratively with us, definitely in the astrobiology realm. Okay, quick breath. Get too excited. So these are some examples of the life we're actually finding in the deep sea sediments, and are inhabited by the three major domains of life. And this kind of illustration is actually from one of our graphic novels, and it demonstrates the rocks eating and breathing the rocks, the microbes are eating and breathing the rocks. So why do earth scientists need our biologists? Because we have these science plans here, and I've got a few copies here if anyone wants to take them home. And they're also available online and freely downloadable. So we're currently in this decadal plan right now. And these are our four main key objectives of the deep sea drilling program. As you can see, Biosphere Frontiers is one of our main objectives. Going forward, so this one ends this year. Going forward, we have a 25-year science framework. And again, our deep biosphere is a huge priority for this program. And again, we're just not having that engagement, definitely in Australia and New Zealand, with enough biologists. And we have a lot of resources, and we do try to attract a lot more because we have so many unanswered questions. I definitely recommend to read the book because nearly every single page is just questions. We have no idea. This is it. This is our limit. We need more people researching. And as you can see in this kind of diagram here, everything's connected. Definitely that biogeochemical cycle. If we want to understand climate change, we need to understand the biosphere. If we're going to do policies to put carbon into the earth, how are the microbes going to eat it? Are they just going to burp it back up? Is it going to be a failed? Yeah. So yeah, just moving on. And some examples of communities IODP is interested in. So these are not exhaustive, but just a few. So we're interested in coral reef communities. We've just drilled off the shore of Hawaii on some drowned coral reefs, led by University of Sydney, Jodie Webster. You can see the riser drilling there. So it's anchored, it can do a bit of shallow water, but it can go really deep. So we have microbes in sediments we look at. Cold seeped communities, we're interested in microbes in the crust as well. In the hard rock, we're looking at bottom dwelling communities as well. We're interested in hydrothermal communities. So Gordon will talk a bit more about these hydrothermal kind of serpentinite communities, the photic zone communities. And again, we do the borehole observatories for long-term studies. And that's not exhaustive. We just don't know enough about this ecosystem and how it interacts. So we're very open to other studies. And for example, I'm about to go through some major IODP subsurface life discoveries, but I just wanted to demonstrate that bottom one there. So we actually sailed someone from the CDC twice. He could go on any research vessel and collect his samples, but definitely the Geordie's Resolution is a famous floating laboratory. You have a biology laboratory on board, so you can collect your samples and do your research there and there. So You can do things outside of expeditions on transits too. So this person did global dispersion of disease-causing microbes by dust storms and its relevance to agriculture. So very cool research. But some other major IODP subsurface discoveries have been that we've discovered that the deep biosphere is more alive than we thought. We've found the bottom, hopefully the bottom of the biosphere, long lifetimes of enzymes that break down carbon. We've also found the composition and structure of subsurface communities, and not all of them. We still got to do more. So for example, the kind of archaean-infecting viruses are an order of magnitude more abundant than the cells that might actually host them, which is very surprising. Another example is oxygen and organic carbon concentrations are key environment factors for defining taxonomy, composition, and diversity of marine sediment communities. Astrobiology has been a huge focus, and it's been predicted to be a model for life beyond Earth. Definitely these microbes that eat and breathe the rocks. The contamination and sampling have been a key factor as well. We work with a lot of programs to understand what needs to be done and how to reduce that contamination. So we can talk about procedures and significance of biogeochemical carbon cycling of the deep biosphere. Okay, I'm getting close to my time. And Linda and Lucinda will talk a bit more about ancient DNA, improving our understanding of past climate change. So I just wanted to do 2 quick examples, going into a bit more detail, but there's many we could talk about. So just looking for the life, how deep did we actually find life? So in 2012, on expedition 337 off the shore of Japan, we found life down to 2.5 kilometers. And that was excellent. So it was a cumulative evidence. So these are all our IODP sites from 56 years of drilling. And that yellow one there is actually the deepest hole we've ever found life. And that was buried in some coal-bearing sediment. So in the past, it was actually a swamp environment and it had been buried deep. So there was a lot of food for these microbes to eat. So that's our current limit of life, how deep the biosphere can go. How hot is too hot for Earth-style life? So again, in actually Japan with the Chikyu, we found this amazing profile. So in Japan, at this trench is a unique opportunity for a geothermal kind of gradient. So it's very rapid, it gets hot very fast. So we want to see how hot is too hot. We could drill quite far down. So in 2016, we went down to that limit of life of 120 degrees Celsius that we know for the hydrothermal vents. We have those hypothermophiles. They can survive, but they're in a nutrient-rich water. Can we find this in the sediments the same? And we did. So we found all the way down to that kind of limit of 120 degrees Celsius in the sediments, which was about 1 kilometer down, and there was still little organisms. But it was quite interesting stratification of the different organisms as you go down. And I just want to end my part just on 2 slides, just explaining a little bit more about the ANZAC office. So what we look after, so over at RSCS, we cater for the chemists, scientists, biologists, modelers, and the climate scientists. And we actually look after all these universities here marked. Currently, we're funded at the Australian side's funded by the ARC. LEAF grants, but we just got awarded NCRIS funding, so we'll be switching over this year. And also we'll be getting ICDP membership, which is international continental drilling too. So we'll be doing land to sea, and we can be doing these big projects as well. So my last slide is some opportunities. So we provide opportunities to sail on these expeditions and ICDP. We generally pay for everything, so the travel costs, berths costs, and we give you about 40K to do your post-cruise research. And you're working with international teams. I've sailed for a year collectively of my life on the Lease the Geordies resolution, so I can definitely talk about that, about expeditions. We give funding to support the study of previously collected samples. We have a microbiology store as well, amazingly curated, big libraries of mud. We normally give small projects about 20K, For analytical funding, we're hoping to open a next round in mid-year. So be on the lookout for. If you're part of one of those universities, you're eligible for those funding. And we're actually launching large projects, how we get international teams together, go to core repositories, working on borehole observatories, and actually answering questions, because we've got a lot of mud, a lot of sample. There's new techniques coming in, and it will be great to engage more scientists. We give access to education and outreach. So I've requested some Chicxulub crater, the dino killing impact. I've actually requested some of that for teaching, so kids can actually touch the impact crater, and vice versa. You can do loans for up to 10 years. So that's all free, and they ship for free as well. Great resources. You can submit proposals to new drill sites, serve on international panels, and we're also, I mentioned that we're getting incoming continental drilling membership for Australia. Okay, so that's me done. Again, we'll wait till the end to have questions. And I'll hand over to Gordon next. Hello, Gordon.
Hey there. Can you turn off your share screen?
Oh, yeah, sorry.
There we go. Now I see it. Mine's coming back. Go. Is my screen coming through okay for people?
Yep, that's good. We're just working on the sound. Oh, sorry.
Good to go?
1 second. There we go.
Alrighty, good afternoon, everyone. Going to highlight some of the work that took place on board the Geordie's resolution for Expedition 399. So it's the building blocks of life, that the rocks that we ended up drilling are what was likely on Earth at origin of lifetimes. And so, you know, hydrogen production from serpentinization, methanogenesis, methane oxidation, the whole wide range of carbon cycling processes can be happening in this environment. And there we go. So I'm going to highlight a little bit of prior research that we've done, and then as an introduction to the IODP work that were there. We work on kimberlite weathering and carbon sequestration, and so peridotite that we were targeting at IODP are basically rocks as food, as Sarah was highlighting. We're interested in weathering of these materials because it promotes mineral carbonation, so carbon sequestration, and serpentinization at low temperatures as a hydrogen producer for life, but also potentially for electron transfer, like it's blocked correctly. And so we'll look at life in the deep subsurface, both land and sea, and then get on to the Geordie's resolution and some of the drill work that we did there. Now we'll start with Diavik. It's in Northwest Territories, Canada. This is a diamond mine. And in this environment here, you can see kind of a darkening along the walls of the mine pit. And when you zoom in to where water is encountering kimberlite, we have biofilm. And certainly it's driven by photosynthesis in this, but it's not just photosynthetic organisms, right? It's a whole ecosystem of organisms. And in this case, they're growing on water rock interactions. And we're interested in this weathering process that happens in nature. Now, a cartoon on the left of kimberlite explosions. They come from deep within the earth, hundreds of kilometers, and they're explosive. We've seen charred wood, bones, things like this that are in the kimberlite pipe that were plants and animals that were there when it blew. Now, after the kimberlite explosion, the upper surface, right, will be exposed to rain and water, and it will weather. And it will produce something like this. This is the Kervla yellow ground. It's a mine in South Africa. And weathering here sequesters a massive amount of calcium. One of the other things that was exciting about this sample is that we could extract DNA, and the ecosystem here was consistent with serpentine soils, and it also contained living organisms, that it wasn't just a geologic time phenomena, that these processes are still happening today. And so for the microbiologists in the crowd, on kind of the your left view of the image, we've got kind of the typical Colobacter that you'd expect from low nutrient environments. We've got tight helices, and the one on the right that we've affectionately referred to as the corn cob, you know, I've been through Berge's manual of determinative bacteriology, and you just don't see this organism out there. So there's a lot of unusual organisms in these environments. And these things are growing on kimberlite, right? We give it kimberlite plus water, that the organisms have to get everything they can or they require from the rock. So in the Kimberlite De Beers project, our assignment was to sequester carbon. And so the control in the background there, just behind the plants, is kimberlite. It's been kimberlite for 3 years, and it hasn't really changed. In the system that we inoculated, these plants have developed. Now, we didn't plant anything. This is just seeds that have blown in, landed on the microbially weathered kimberlite, and basically signaled to the plant that you can have a go here, right? That it's a good enough soil for these things to grow. Now, if you dig into where the plants are, you can see that there's extensive weathering compared to the surface material, breakdown of rock and formation of secondary minerals. If we zoom into the cement in this material, we've got millimeter scale calcium carbonate forming, just like we saw with the yellow ground. If you do a weak acid etch on these carbonate grains, you'll expose clays and bacteria. Again, the DNA here consistent with the serpentine soil. And so we're sequestering carbon in a stable form by weathering ultramafic material like kimberlite and like peridotite in the ocean. The other side of it is this low temperature serpentinization. And this fits into Sarah's, you know, kind of electrochemistry that's in there. That clays can have organics, that's food. We've got nickel sulfide that can be food. And serpentinization adds food to the ecosystem. And this is one of the things that will be active in the Atlantis Massif. So in serpentinization, we've got the mag sulfate, so we've got forsterite, and we've got the iron silicates, the phthalate, and it hydrates with water and it makes hydrogen gas. And so this is an energy generator that should be active in the ecosystem. And now if you notice the connections here, these really don't look like capsule. And there's a lot of interest in microbial ecology and electron transfer, the idea to pull electrons or to dump electrons, in our case, in rock systems. What we've seen in our serpentinization process, take a peek here, that we've got bacteria that have literally plugged themselves into the rock substrate. And wouldn't it be interesting, we haven't demonstrated this, if they can pull electrons straight from the rock, they'll produce some protons as byproduct, but this would be a mechanism to drive that low temperature serpentinization. So let's take a look into the deep subsurface on land. The photo on the right, you're staring down a mine adit that's 2 kilometers deep. You go down 2 kilometers, you walk across a few hundred meters, and then you take another cage and you go down another 2 kilometers or so. And in the Driefontein mine, we've gone down 4.2 kilometers below land surface. And we were interested in life in extreme environments in this project. So here's a drill hole. You're in the mine. We're 3.1 kilometers down. And the drill hole with the arrow there that you're seeing goes down another 600 meters. And we found a very diffuse, low biodiversity ecosystem that the molecular guys in the group said, we have an ecosystem of one. That it's a sulfate reducer, it can grow on hydrogen, it's capable of sulfate reduction, it has hydrogenase, so all the serpentinization reactions can feed this organism, but there's not a ton of biomass down there. And so there's real questions on what signals tell a bacterium that it's okay to divide and produce a competitor that's similar to you. We've also been in the Northern Platinum mine. Platinum mines are ultramafic, and so the ultramafic rock fit in with the same serpentinization process. And in this environment, we had methane being produced and coming out into the seep. So we're getting the same kind of biofilms from water-rock interaction where there's no photosynthesis, right? This is just bugs growing on the rock in this environment. And one of the organisms that kind of came out of this one was, it's strange, it's a branching bacterium. It looks like it's gone through the Plato extruder, giving us the little ridges there. The student who had originally worked on these samples had shown me these thin sections, and I just told him to do it again. And he said, no, I've done it. I've done it a few times. It's real. It's not a fake thing. So we got a strange organism here that's increasing its surface area to volume ratio by producing this kind of star-like structure in cross section. So let's move on to the JOIDES resolution. So this was in the Azores, kind of the day before departure, and we had an 1,800 kilometer trip ahead of us from leaving port. So here's we get down to the site. We're at the Mid-Atlantic Ridge. It's where the plates are coming apart. We've got the Atlantis fracture zone. The massive itself is basically a 4-kilometer mountain on the ocean floor, where the abyssal plain there is at 5 kilometers. So the water that we are drilling was only on the order of kind of 700, 800 meters deep. And we were interested in the relationship between the Atlantis Massif and Lost City. Now, if you haven't seen Lost City or pictures of it, you need to Google this. This is the kind of thing that you get. The carbonate spires on the right are the size of skyscrapers, right? This is a huge structure. We're on the order of 40 meters in height in this environment, and it's a low temperature hydrothermal system, kind of 80 and 90 degrees C. Now, we were drilling up on the top left there, the top platform, and our goal in this project was to drill down 201 meters, which was the previous record for ultramafic rock. And so you can see the scale bar on the bottom left at 50 meters there. And so we were trying to look at the plumbing network that was feeding the Lost City hydrothermal field. And so that was kind of our goal in this process. So we've got a couple of holes here I'm going to talk about. So hole 1601, that was within 800 meters of Lost City, and our initial goal with the 1601A was a preliminary drill hole, and we were hoping to get down to 50 meters to see what we would get. We'll come back to 1309D in a minute. So here's kind of the drill rig and the drill core coming in day and night. The plan that they had originally is that they thought they would be able to drill 1 meter per hour, and so we would get drill core coming up every 5 hours or so. What in fact happened is we were getting drill core every 70 minutes, and so we had to process samples within that 70 minutes before you got the next core on deck call to go up there and get your next sample for the project. So we eventually at one point started running out of supplies on the ship. We were getting so much core. Good problem to have. So here's our drill core. It comes in. At that point, it's moved into kind of the washing and cutting lab. This is Billy Brazelton. He was my bunkmate. So when he was at work, I was sleeping, and vice versa. And so as a microbiologist, we were given first crack at the core, and were allowed to collect whatever sample we wanted under the watchful eye of the geologist. So we got sterile Teflon here. You grab the sample, and then it would go down into the geomicrobiology space. And so we've got kind of a benchtop laminar flow system. The boxes just behind it there, not sure if you see my arrow, were kind of washed and sterilized, methanol sterilized. And so the first thing that we had to do was recover the rock from the inside of the core. Now we had fluorescent tags that were put into the drilling fluid, and so the outside of the core that came out on deck would have had this fluorescent tag all over it. And so the first thing we had to do was chip off the outer half to 1 centimeter of the sample here. My kind of lab partner, Feng Ping, she was spectacular at just kind of the right touch to knock off the outside of the sample. And with the exception of a couple of samples that were made of soft, friable serpentine, the dye did not penetrate the inside of the rock, that we could demonstrate that the outside was contaminated, and our chipping strategy removed it. After Feng Ping had done that, it came to me. And my job was to take the rock and crush it. And we didn't let the geologists in here because they just looked very sad as we were busting up the rocks for them. But they got lots of material for themselves. So anything that didn't go to biology was slabbed, half went to storage and half went to description, and will be available for science. The geologists were really interested in these kinds of samples. We've got serpentinized material, right? So the hydrogen generation should be going on. And we've got Harzburgite and Dunite, which is rich in olivine. And the olivine is that forsterite phalite that's key for serpentinization processes. Now, I really lucked out on this trip over these samples. Now, they were within the surface, kind of within up to kind of 30 to 50 meters of the ocean, and they were weathered. And so the geologists said, oh, they've been exposed to seawater and circulating fluids. They're ruined. They're not any good to us. And my microbiology colleagues who are interested in life in extreme environments and boundaries of life wanted deeper, hotter, saltier. And so anything that had interacted with seawater was of no interest to them. So I was the only person on the ship that wanted these samples. And if we just kind of zoom in on one of them there, you can get a closer look. We've got the cross-hatch pattern of serpentinization. We should have nickel driving that reaction. The veins here are carbonates, right? So we're sequestering CO2. And so there's not a lot of DNA in the material, so we're doing multiple extractions. We're going to do the acid digests of the carbonate grains to see if we're finding interesting bacteria in the sample. We were awarded synchrotron time, so March 8th we'll be putting, I expect this sample, it's our favorite, into the beam for the first time to be able to map metal distributions in this environment and hopefully relate that to life processes. One of the challenges we had on 1601A is that the drill bit jammed at 60 meters. We were hoping for 50, but everything was going just so easy. They said, well, let's just keep drilling. And at 60, we were stopped. I came up, people weren't working. They were just kind of hanging out. And it's like, what happened? And they said, oh, we're stuck. And it would take a day and a half for us to get unstuck from the site. And it would involve dynamite to break the joides off of the ocean floor and release us so that we could continue our work. Now, on the left there, you've got hole 1601A. That was our 60-meter hole. We got great samples, but we were stuck there. We went on to 1601B, and it was a disaster. It did not go well at all. And the plan at that point was to go to 1309D. We did eventually get a 1601C hole, and that was where we went down 1.265 kilometers, right? So we were well over a kilometer deeper than anyone had ever gone before in peridotite. So it's a spectacular suite of samples. I'm going to finish up with U1309D. It was a 1.4 kilometer drill hole. It had been drilled 20 years ago. It had a reentry cone. And so it was considered an ideal site to go back and look to see how the system had changed in that 20 years. Now, one of the first experiments we did was Jeff Wheat. This is a 10 times 1 meter, so a 10-meter long water sampling system where the collection trigger was going to go based on temperature. So he could set it for 90 degrees C or 100 degrees C or 120 degrees C, any temperature he wanted. And so they were interested in going from above to below the temperature limits of life in this environment. Now, this rig that he developed took years to design and build and test. On the ship itself, it took the full 2 weeks for Jeff just to assemble this in the lab first and then down on the rig floor. And he was getting one shot at this on the whole trip. He's going to be there for 10 weeks. And he has one try to see if he can sample water based on temperature. So what could go wrong? Well, 1309D is not vertical. It's off by about 1 degree. And you wouldn't really think that's a big deal. But what in fact happened is that as it went down the hole, it started scraping mud or rock from the wall of the borehole. And as it went down 1.4 kilometers, it picked up an awful lot of crud that's seen here. I got up, I came off, you know, kind of woke up, had my breakfast, and everyone's talking about Jeff's mud. I'm like, what? The guy's a water chemist. What's Jeff's mud? And so I went up there and he's, look at all this crap. I gotta chuck it and clean everything up. And I just said, don't touch it. Don't let anyone touch it. And I ran down to the lab because to me, the sample was kind of one of the most beautiful things I'd seen at that point on the cruise. So an hour later, each of the mud piles had turned into 3 samples, one for SEM, one for molecular analysis, and one where I just added seawater, right? That if the ecosystem down there is exposed to seawater and that can enhance growth, then we're not going to give them any food other than, again, the mineral substrates that are there. If you wash the black mud off of it, we found that all of the rocks in there were coated in iron oxide, and we had just this small benchtop SEM. That was just a fantastic instrument that could withstand all sorts of vibrations and everything going on with the ship. And so when we looked at the iron oxide, we found things like leptothrix. You can see all the long filamentous cells. Wherever there's breaks, you can see there's a core, and so it's full of life in this environment. There were larger round things like in the middle, and we've got these templated structures. I don't know what they are, but we've got it back at UQ now, and we've got better microscopes. And so looking forward to having a really close look at the inside of this, as well as making polished sections to go inside the iron oxide coatings. One of the presents I got before leaving the JR is that with the mud that was left over, I'd set up a few extra enrichment cultures just to play with it and see what was happening. And so within 6 weeks of having these samples, putting it back under the microscope, we have cultures. So we've got evidence that there are bacteria growing from that 1309D, you know, kind of mud that we're scraping off the side of the borehole. So we're really interested to see what we've got here. The DNA concentrations are reasonable, and we've got to pursue both that as well as the imaging. At this point on the JR, and like today's seminar, I can just put my feet up and relax, and we'll have questions later. So thanks.
Thanks, Gordon. That was fantastic, as always. Just for the narration of time, we'll move straight on to Linda and then on to Lucinda. Thank you.
Hi, everyone. I'm Linda. This is Lucinda. She will just start very briefly.
Hello, we just wanted to say we're calling from Nipalina, Hobart, and we acknowledge that we're living and working on Aboriginal land and acknowledge that sovereignty was never ceded.
Thank you. So I'll start. I'll be not moving on. Sorry. Yeah, okay, great. Yes, so I'll be talking about an ancient DNA record that we generated during IODP Expedition 382 that went very far back in time. But to give you a bit of an overview about ancient DNA first, ancient DNA is DNA from dead organisms. It's very fragmented and degraded and only occurs in trace amounts in the sediments. It's been used to study human and megafauna evolution since more than 30 years now, but the application to marine settings is still relatively new, and so we're constantly troubleshooting and finding out new things at this point in time still. Modern DNA is very different because it is from living organisms, very intact, very abundant everywhere around us. The techniques are much better established for modern ocean settings, and there's, for example, some really big ocean sampling programs out there that collect DNA samples all around the world almost every day. And that's really important for our work because they're generating reference sequences that we can use to compare our data to. Otherwise, we would have no idea what we're looking at. So the idea of ancient DNA is that we can reconstruct past marine communities. And that's great. We can look across the food web. We can look at bacteria, viruses, phytoplankton, zooplankton, even fishes. But there is some caveats and some limits to this. So, for example, we have to be very, very careful with contamination and making sure that we're not introducing any modern environmental DNA signal into our samples. So what you're seeing on that photo here is a very, very bad idea when you're sampling for ancient DNA. We should never take the samples with our bare hands. So, yeah. And another thing we want to look out for is that we are actually authenticating the DNA. And there's a couple of analyses that we can do to show that it's free of contamination that I'll talk about in a few minutes. So as I said, the data that I'll show and some pictures that I put in this talk here is all from IODP expedition 382, Iceberg Alley and Subantarctic Ice and Ocean Dynamics, which was an IODP expedition in 2019. And so on this expedition, we had an outreach officer, Marlo Garnsworthy, who made this really nice animation about the region. And it shows you here on the left how icebergs are being pushed off the Antarctic continent into the ocean. They travel around the entire continent and they end up in this top left corner here. I'm not sure if you can see my mouse, but that's where all the green dots are, which is Iceberg Alley. So all the icebergs pretty much end up there. And by coring in this location, we can reconstruct Antarctic ice history from the entire continent. So that's why we went to this particular region. To show you in a bit more detail, on the right here, you can see a map of 3 stations that we caught during this expedition. So there's U-1534, which was on the Falkland Plateau, and U-1536 and U-1538, which was in the southern and the central Scotia Sea. The site names are basically running numbers of stations that the IODP has drilled before. That's why they've got these funny numbers, and there's lots of them. Yes, so we went on the JOIDES Resolution. Here in the middle, you can see a picture of the drill rig. I was standing in front of it, so it's huge. And in the middle is where the sediment cores are brought up into the ship. So they come up vertically in 10-meter pieces, and then we flip them on the side, and they get transported in this lab that you see on the bottom left picture. So they get shuffled in there. When we're taking ancient DNA samples, we have to be a little bit thoughtful of which coring technique we use. You can see on these pictures here that piston coring brings the nicely layered sediments that we would prefer for this type of analysis. So there's other coring systems available too, which you could go deeper into the sediments, rotary coring, for example, but there's a little bit more disturbance of the sediments. And so we limited our analysis or our sampling to the piston coring. There's one caveat with piston coring. It's a hydraulically triggered system, which basically means that we are pumping drill fluid, which is more or less nasty seawater, towards the triggering mechanism. And there shouldn't be any way that the seawater actually gets into our core, but of course we want to make sure. And so we can track this by infusing the drill fluid with a chemical tracer that we can then test for along the length of the core. And so this is a technique that's been regularly used for microbiology sampling on board the JR. For our expedition, it was the first time that was applied to ancient DNA analysis too. So when the cores come on deck and get brought into the lab that I just showed you from the outside, here on this first photo, top left, you can see the lab from the inside. So we have the entire length of 10-meter core in front of us. And the first thing we do is to decontaminate it with bleach from the outside. And then we decontaminate all the core cutting tools as well with bleach and ethanol. And we cut the core into 1.5 meter sections. And then we can sample from the bottom of each section. We could sample from the top too, but we just did the bottom of each section regularly for ancient DNA samples in the center of the core and for the chemical traces at the same depth. We take lots of environmental controls also for here. So here in this expedition, we took air controls, just holding tubes up, took some swabs of the environment. We were working in drill fluid, and that's to be able to analyze basically the environmental signal that we're sampling in and can compare that to our ancient samples. We collected a total of 80 samples on this expedition using this particular sampling techniques, and then shipboard analysis and also post-cruise analysis confirmed that these were up to about 1 million years old. You can see on the pictures also our precautions to try and not introduce any contamination from ourselves into the core with lots of PPE. So if you want to make enemies on an expedition, you say you want to sample for ancient DNA, and yeah, everybody has to wear face masks and all of that. But it was actually a lot of fun, and I had a lot of help in the end. And you can see Sarah here, who's been really helpful too in the top left picture there, helping with the sampling. So it was really great. So on the ship, we freeze the samples and then they are brought to the lab. And you can see our paranoia about contamination gets worse and worse the further we go in the process. So in the lab, we're wearing this full PPE with full coveralls and visors and 2 to 3 pairs of gloves to not introduce contamination. So the labs that we're working in, they are ancient DNA facilities, which means these are regularly cleaned labs under positive pressure so that no contamination can get into the samples. We introduce more controls during the extraction process, so extraction blank controls, so that we can assess whether we might have any cross-contamination during the process. And all these protocols to extract DNA and to make sequencing libraries are relatively well established now. And so we followed those protocols that are optimized for marine eukaryote ancient DNA. Then we sent the samples to sequencing. And what we're aiming for here is to do a metagenomics approach. So we do metagenomic shotgun sequencing, which basically means that we're randomly sequencing all the DNA in our sample. And that has a couple of advantages. First of all, we're keeping our community data in proportion, so we can look at that at the data semi-quantitatively. And also this technique preserves the size variation of the DNA fragments we have in our sample. And that's really important for authenticity assessments. So there's different techniques we can do to assess whether we have authentic ancient DNA. For example, if we go deeper in the core, we would expect the DNA fragments to be more degraded, more fragmented and shorter. So we can test for this. But what is a much better technique is actually DNA damage analysis. So if you imagine a DNA fragment, the ends of the fragment are exposed, and this is where damage normally occurs. And so this is something we can test for bioinformatically. And that's exactly what we did with the IODP Expedition 382 samples. We used a program called HOPS for this analysis and looked at the damage patterns in the marine eukaryote DNA and found that it increased with depth. So this was a very good indicator that we're actually dealing with ancient DNA in this case here. We did a couple of correlation analysis with other parameters that were measured during the expeditions. I won't go too much into the details here, but one point maybe to note is that the maximum damage we find in these samples in eukaryote is around 30%. There's not much data we can compare this to. There's one core from Tasmania where we found a similar upper limit of DNA damage. But it's the only core we can compare to, and that's what I mean at this when I said at the start. Sometimes we're hitting the limits because we just don't know many things in this field of research yet. There's so much more to be explored. But then, of course, we want to know, what are these sequences? What's the community looking like? And so when we look at the community data, we basically get these trees, but they very much depend on which reference database we use to compare our data to. So for our Expedition 382 shotgun data, we basically wanted to see what's the community change through time. And so we use taxonomic marker genes to compare databases of taxonomic marker genes to compare the data to. And there's two different databases that are commonly used for marine phytoplankton, which is in short the SSU and the LSU. And we merged both to be able to get more sequences aligned because we're dealing with relatively low sequence numbers here. And then this is some of the results. So I've plotted the eukaryote relative abundances here with increasing age at the 3 different stations. And one of the first results that was really exciting was that the deepest samples here that were about 1 million years old actually had some eukaryote ancient DNA preserved, which was really exciting. Because before this study, most of the studies actually, had timeframes of maybe 10, maybe 20,000, 30,000 years. There was one study that also looked at 140,000 years, but 1 million years is a very significant extent into the past. And it means that we can look at community changes using this technique over multiple glacial interglacial cycles, which is great. And then we also found some interesting patterns in this data. When you look at the U1538 side on the right, you can see at about 14,500 years, we start to see this increase in the relative abundance of the olive-colored taxon, which is diatoms. And so, it's the relative abundance of those diatoms increased from less than 10% to more than 50% at about 12.7 thousand years. And that was a time that experienced some significant warming around Antarctica. And we did some further analysis into photosynthetic genes using the same data and could determine there was most likely a quite significant restructuring of the marine food web happening. But in the interest of time, I will stop here. I just want to say that this data set has been fantastic to work with over the last couple of years, and it keeps on giving. So there's multiple projects here that use Expedition 382 data, partly in the research. There's students working on this and postdocs, and there's all sorts of projects, for example, focusing on diatoms, on Antarctic krill, on radiolarians, or on improving the bioinformatics pipelines that we have. And with this, I'm at the end of my talk, and I will hand over to Lucinda.
Should we do the swap of the laptops as well?
Yeah, I'll stop sharing.
Can you guys hear me? Wait, I'll try and be speedy. So I'll get into it. Oh, here we go. You just put this in. Hello everyone, I'm Lucinda, and I am going to be sailing as a microbiologist and a geochemist aboard the IODP Expedition 403, which will be the last hurrah aboard the JR. And so from June this year, I'll be spending 2 months in the midnight sun, working 12 hours on, 12 hours off, up on this voyage to the Arctic, where we'll be looking to reconstruct the past environmental history of the Eastern Fram Strait. And speaking of straits, I'll get straight into it. So we're going fairly far north. We're going to the 79th parallel, which I was looking it up on Wikipedia, and if you believe Wikipedia, apparently only 186 people live further north than this point on a permanent basis. But I fear this may increase with climate change because the Arctic region is actually warming at about 4 times the rate of the global average. And with this trajectory, we're looking at potentially ice-free summers in the Arctic Ocean by mid-century, when I will be middle-aged, which is quite frightening when you really think about it. So the area that we're going to, I don't know, can you see my mouse? I don't know. Oh, cool. We're going to the Fram Strait, and if you remember from that first ocean map that Sarah showed, you can see how the Arctic Ocean is landlocked, and actually the only way that water gets in is really through the Fram Strait. And you can see this red line is the warm water that is funneled from the more southerly latitudes up into the Arctic. And where we're going to be sampling, we're going to have a nice sort of north-south transect along this warm current that's coming in. And this current exerts a significant control on how much ice is going to get melted in the Arctic Ocean. And that is obviously important for things like sea level rise, but it's also important for controlling how much solar radiation is reflected straight off the ice, which is pretty good, or how much is absorbed by an open ocean, which is not so good climate change wise. And it's also, this channel is a really important part of the thermohaline circulation, which drives ocean currents and climate patterns all around the world. So long story short, what happens up here affects the entire world. So at each of our sites, we'll be drilling into sediment that has been accumulating for millions of years. So we can understand how this system behaved in the past, with the view that this will inform future predictions for this region and the rest of the world. And at one of the sites, we'll be drilling more than 700 meters into the seafloor, which is a huge operation. And for context, the experience that I have is drilling 7 meters of sediment through 90 centimeters of water on a little lagoon on Kangaroo Island. So this is a big step up in terms of logistics for me. So very exciting. So we'll be traveling potentially back in time 7 million years with these records. And you can see from this figure here along the bottom, each of these black lines is the cores that we're expecting to get from each site, and how far back in time we're expecting them to span. Just on the right here, we've got 0 million years, so that's present day, and then going back all the way to 7 million years, hopefully, fingers crossed. And what you can see from this part of the figure up here is it covers some really interesting periods of Earth's climate. And we've actually got, you can see in the yellow highlight, that's periods of time that are analogous to the warming that we're seeing today, and potentially future scenarios that we might be going into the future. So it's really important we can look back in time and see what was happening in the Fram Strait, and how the warm water being funneled up through there might have been affecting how much ice was being melted and the global ramifications of that. But as a microbiologist, what I'm interested in is how the marine microbial community is going to be responding to those changes, and linking that back to how that would affect biogeochemical cycles, and then how that would go back and then affect climate. And so you can see how the biology is really important to sort of bring things full circle and to be able to draw those connections between all these different fields of study. So one thing that we're hoping to dive into, quite literally, in this expedition is how far back in time ancient DNA might be preserved in the Arctic. And so Linda was saying that some of the records from the Antarctic go back 1 million years. We're interested to see if we could go past back 2 million years. I'm not sure, we'll see how we go, but I'm working with Doctor Stein, so reference to Doctor Who and time traveling. Anyway, moving on. So yeah, that's something we're interesting to see, and it's a good place to test this out. The preservation of DNA up in the poles and really deep in the ocean is thought to be quite good. Something else that I will hopefully be addressing in my PhD is exploring the potential for ancient RNA, which hasn't really been investigated at all in this context. And traditionally, RNA has been overlooked in an ancient context because the molecules are much less stable than DNA. But there's been some recent studies that have surfaced lately that are showing RNA can actually preserve in really good conditions. And it's something that would provide really interesting insight into how organisms are regulating their genomes, because RNA is so important in the regulation of the genome. So we could look at how genes are regulated in response to environmental pressures on evolutionary timescales. I don't know if it will work. Stay tuned to find out. I think I'll leave it there, and thanks for listening. And here are some ways that you can follow the journey. There's Instagram, Twitter, a website, and there's going to be a film crew traveling with us, which is very exciting. So I think they're going to put out a documentary called The Time Travelers. So look out for that. And I'll hopefully be writing some articles in Cosmos about what I've been doing. So yeah, thanks for listening, everyone. Oh, that's a spare slide.
Amazing work, Lucinda, and all our speakers, Linda and Gordon as well. Thank you so much for sharing your insights and broad context, even the microbiology fields themselves, let alone the frontier science of every expedition. Do we have any questions online or in person? If not, feel free to contact these people anytime and ask them. We got one up the front. You might want to come up or...
Okay.
Yeah. You're at the
I love the talks, really great. I was wondering about the second talk. You mentioned the biofilm formation of bacteria on rocks and how that might have enabled them to have special function. I was just wondering how is that just done by microscopy, or do you actually, can you figure out which species are embedded in the biofilm and how it's structured and what the functions of different bacteria are?
Yeah, so I've got a student who's been playing with kind of biochemical pathways, KEGG pathways. So, taking a metagenomic approach similar to colleagues there, right? So we get the kind of the sum of the ecosystem. So we've got Phil Hugenholtz in the Australian Centre for Ecogenomics. So we have students that go over and join his group there for training in looking at sequencing. For the mines themselves, these are all over the world. You get mine slimes wherever there's water rock interaction. Now certainly there they're in the mine, right? So they're exposed to oxygen, right? And so we had we just assumed that they were weeds, that there wasn't really much interest in them, and it was kind of years on where kind of the idea that we've got these unique ecosystems. Now what's interesting both in terms of the kind of the mine slimes there and the cultures is that we're adding sterile water, right? We're not adding any nutrient. There's no nitrogen, there's no phosphorus, there's nothing. And so they've got to weather things like apatite to get phosphorus. There's often trace organics, and so they'll eat whatever they can find.
Thanks, Gordon. We have a question online. Craig, unmute yourself and you can ask away.
Right, thank you. That was a fascinating seminar. I apologize, I'm not there in person. Can you hear me okay? Uta nod, Uli nod, yep, okay. So I'm fascinated and frankly astonished by the paleogenetics and how far back you can go here. That's very, very exciting. So I'm wondering, can this be used to inform understandings of model circulation through Pleistocene cycles, or do we still not understand enough about the link between the composition of the biota and oxygen availability or other factors relating to ocean circulation?
Should I have a go?
Probably like you, Linda. Could you understand the question?
No, it's all good. But we have two laptops right next to each other, and they have a terrible feedback. Yes, I think yes, that's exactly what we would hope to do. To look into the past signals to inform what's happening in the ongoing climate change. I guess what's critical is a good sampling resolution through times that have experienced previous warmings, maybe even warmings that were warmer than what we're expecting now, to look for those kinds of signals, and then have a good set of metadata collected on the same expedition to be able to assess the environmental variables that are or the factors that are happening at the same time. And normally I would think that an expedition like the IODP expeditions, also other expeditions, they have such broad teams of researchers on board that that is actually possible. I guess you have to target the right time periods while you're on the ship, which you would sort of have to do with the first set or with the first set of data. That might be limiting. There's a danger to be off slightly in time, so a little bit of close interrogation of the data you can collect on the ship already is good to guide the sampling a little. I'm not sure if that answers your question, but
it does. If I can just follow on, because it also depends on where you are, of course, in relation to expected circulation patterns and overturning patterns of upwelling, downwelling, and so forth. So can you use ocean circulation models, I'm talking about three-dimensional ones, to try and predict where best to sample to detect change?
I guess, yeah. I don't see why you couldn't use a model to find some good sampling locations and regions. The additional factor to consider, though, is DNA preservation. So even though you find potentially a really ideal site to sample, the preservation might not be as good. That is something that, by looking at different data sets from all over the globe, basically, but mostly around Antarctica, different regions have very different preservation levels.
Yeah. All right, thank you.
Yeah, I can add a little comment on that too. So there's definitely for the Antarctic region. So there's this great group called SCAR Instant. So it's the Scientific Research Committee for Antarctica Research. And it really is pretty much a 50-50 community with the modelers, the ocean and climate modelers, with the paleo community. And it's the first time. So the big issue with using ocean circulation right now, while we haven't really considered it too much, is we speak two different languages, even oceanography and the paleoceanographers. So we're finally overcoming that. We've realized we've been studying separate, same with the biology. So we've got some great biology paper, octopus DNA, back in time. But yeah, that should be, if you're interested in that stuff, definitely in Antarctica. I think they're the group leading, and I think in IODP into the future for the next 10 years, every nation has pretty much indicated that the polar regions, these high latitude sampling drilling is going to be the priority for drilling. So if you are interested in these Arctic or Antarctica regions, and definitely with the ocean currents, we're trying to really match up the modelers with our science. I think it's really great. We should sail you, Craig.
Yeah, I'm not sure they'd let me on board, but thank you again.
Thank you. Do we have any other questions? If not, we'll end it there because we're a bit behind time. Thank you to all our speakers, and thank you to all those who stayed on for 15 minutes extra. It's been absolutely excellent. We've recorded it. We'll put it up on YouTube if you want to follow it later. Okay, thank you. Thank you, everyone. Bye.